compulsory check on the, oops, yeah. Okay, microphone is on. Then welcome everybody back to the live stream again. It's the end of the day, and well, I wanted to do yet another live stream, because why not? Just to wrap up the day with looking at some mighty fine genomics papers, and well, in this case I might be better prepared as last time, and yeah, so I wanted to explore a topic that is well, maybe not as usual, and that is the behavioral genetics and behavioral genomics. And let's see what are the possibilities and what are the papers in this direction. One thing which I'm doing here in the background still, because this thing is with the live audience, as if you of course watching it live, then well, I would just encourage you to say hi in the chat, and also if you have any questions and comments, then of course I would like to see them in the chat. So of course, if you're watching this on YouTube for as well, a non-live section, then well, hopefully it's still interesting for you. And also well, I'm doing here and there these well, live streams, so if you are interested in, if you see me live, or well, maybe it will be more with the more regular schedule, then of course I would be happy if you drop in at any time. Good. So we will do pretty much the same thing as last time, basically shooting off random keywords into Google Scholar, see what comes up, and well, checking out some interesting papers. Not the whole papers, of course, but mostly abstracts, going through the figures if there are any, and the conclusions. And yeah, that would be the thing. So, and yeah, so well, this genomics medieval livestock, it was from last time. Anyway, so what is this behavioral and let's go with the animals first. Okay. Also, if there is actually some suggestion from the chat, I also I can take that as well for a paper. Or what if you have, for example, what is your favorite trait in animals or humans that might be connected to genomics? Maybe there is a paper about it. We can check it out. So just put it in the chat, and we can look at the possibilities. Anyway, so this is an intriguing title: behavioral genetics and genomics, Mendel's peas, mice, and bees. Okay, it's quite nice, quite nice to have interesting titles. Well, it's like in particular on YouTube and other spaces, but it seems that it doesn't hurt if you in a scientific papers as well. So this is in the pretty good place. Well, actually, okay. Mendel's bees is clear. Well, I don't know if you know, but Mendel was also a beekeeper, so the Mendel's bees also make sense. Mice, for the moment, I don't know how it ties in, but let's see. Okay, so Hoekstra and Robinson. I hope it's a good, relatively new 2022 paper. Okay, so the question of heritability of behavior has been of long fascination to scientists and broader public. That's true. It is now widely accepted that most behavioral variation has a genetic component, although the degree of the genetic influence differs widely across behaviors. Starting with Mendel's remarkable discovery of inheritance vectors, it has become increasingly clear that specific genetic variants that influence behavior can be identified. Well, that's really intriguing already. So let's see. Come on. Okay, well, maybe there is a... Okay. This goal is not without its challenges. Unlike P morphology, most natural behavioral variation has a complex genetic architecture. However, we can now apply powerful genome-wide approaches to connect variation in DNA to variation in behavior, as well as analysis of behaviorally related variation in brain gene expression, which together have provided insights into both the genetic mechanisms underlying behavior and the dynamic relationship between the genes and behavior, respectively, in the wide range of species and for a diversity of behaviors. Okay, comment here. don't write such long sentences. This was well, I was almost out of breath here, but definitely, here we focus on two systems to illustrate both of these approaches: genetic basis of burrowing in deer mice and transcriptomic analysis of division of labor in honeybees. Okay, this explains how the mice and the bees come in, and the bees is like just from Mendel's default. Finally, we discussed the troubled relationship between the field of behavioral genetics and eugenetics. Well, tough. Which reminds us that we must be cautious about how we discuss contextualize the connections between genes and behavior, especially in humans. Okay, starting off strong. Hi, Dr. Maher Maz. How are you doing? How was your day, sir? Or maybe it's just still ongoing. Well, it's depending on the time zone you are at. Here it's pretty much ending. My day was great, by the way. Anyway, so when I was thinking about this topic, I was actually thinking about this eugenetics part as well, because this depends how you discuss these topics. It's, well, can be problematic. But anyway, so actually this I would like to also check out this part, what they have to say about it. But first, honeybee is clear how it looks like, but I am interested in the deer mice. What is it about? Deer mice. Oh, cute little mice. It looks like more like a field mice or something like that. Oh, okay. But it's deer mice. Okay, now this is a question if it is a full text is available or not. Very good. Very good. So, okay, so now according to our rules, there is a lot of Mendel's diverse interests, other organisms including house mice and honeybees piqued Mendel's interest, but both biological and non-biological reasons its ability to perform genetic crosses on those species was limited. Instead of this elegant study of the progeny of pea plants that he derived in laws of inheritance. Okay. Well, and with bees it would have been particularly problematic because of their specific structure or DNA or inheritance structure that they are having. Okay, but this is a bit different paper, or at least for structure-wise, than I thought, because this is more like a descriptive type of stuff. Well, let's see. Simple versus complex inheritance. Okay. A pea seed color as studied by Mendel exhibits a simple genetic basis. When a pea plants that generate yellow and green colored seeds are crossed F0, the seeds of all progeny are F1, green F1. When F1 progeny are crossed, the resulting F2 progeny produce either yellow seeds or green seeds, fully recapitulating the F0 parental phenotypes with no intermediates. These results can be explained by a single gene of seed coat color, identified in stay green. That is citation type reference with dominant green and recessive yellow allele. Okay, well, this is just for the inheritance type, and this would be a complex inheritance, I guess. Burrow architecture in Peromycus mice exhibits a more complex genetic basis. When old-field mice are crossed with deer mice, F0, the resulting offspring dig burrows like old-field mice, F1. Like this. Oh, okay. Oh, they burrow. Oh, that's interesting. They do different holes in the ground. Okay. However, when F1 mice are backcrossed with deer mice, BC1, backcross 1, offspring display a continuous distribution of burrow length that spans paternal species phenotypes. These results are consistent with multiple dominant loci from old-field mice contributing to longer burrows. Okay, that is quite interesting. I didn't know that actually the length of the poles and stuff is also, well, it seems it's genetically somehow coded, or this behavior, that how big of a hole in the ground they are digging out. That's interesting. But that's also, then it seems that it's, oh well, yeah, so they say that there is a continuum, yes, and more complex inheritance, simple versus complex genetics, behavioral genomics. Well, this seems like a mighty fine paper to read, actually, like in more detail. Okay, let's see what is the other case study here on the bees. Nursing, foraging. Oh yes, because with bee, I think when it's born, it's kind of is with the, or well, born. When it starts his life, then it's mostly like nursing duties, and then goes out, and then just collects the nectar or whatever. So this is the kind of how much is inside and outside of the hive with the time. And this bees, I don't know what it is. The gene expression networks underline the behavioral plasticity. A honeybee worker will begin its adult life working inside the hive as a nurse, and then it will transition through a middle age phase of tasks before it shifts to working outside the hive as a forager. B. Okay, so this was the first stuff. And the behavioral changes associated with a labor transition are tied to changes in brain gene expression with thousands of genes. The coordinated changes in the brain gene expression result in markedly different gene expression networks between nurse and forager worker bees. Oh, this is the thing in the expression of the genes, and then it's like basically a gene network, and the different genes are getting activated if when it's a worker type or worker kind of duties, and the foraging duties. That is quite nice. Yeah. Behavioral genetics and genomics. Next, behavioral genetics, species 1, 2, inheritance, behavioral genomics, environment. Oh, there is a gene expression variation. It is then probably a genotype by environment interaction, or just plain old influence with the mice and the bees. Okay, so the behavioral genetics and behavioral genomics as complementary approaches. Both behavioral genetics and behavioral genomics investigate the link between genes and behavioral variation. Behavioral genetics, as exemplified in the burrowing mice, seeks to connect inherited genetic variation to behavioral variation. Changes in genetic sequence are represented by different colors of chromosome, pink versus green. Okay, so the genetic sequence green. Behavioral genomics are exemplified by a honeybee. Division of labor seeks to connect changes in gene expression to behavioral plasticity. Behavioral plasticity can be caused by changes in the environment, external, internal, which can lead to, for example, epigenetic modifications. Yellow circle, epigenetic is the yellow circles type. Okay, well, it's just everything is kind of summarized here without changing DNA sequence. I mean, come on, this is a. I mean, I don't think it needs. Everything needs a different color, but well, it doesn't matter. Both changes in the DNA sequence and the environment result in changes in gene regulation and ultimately behavior. So behavior is just gene regulation according to this paper. That's quite a nice turn or twist on things. But then, this comes the difficult question. It's like how much we can regulate our behavior if it's genetically regulated, or I mean, what is the kind of a free will? What is the? Well, that's stupid. No, it cannot be. Well, I'm wondering what is the heritability of the whole thing, and what is the environmental part, and what is the genetics part, and what is the learned part. That is a bit of a good question here. But anyway, so it's nice to see that it's there are indeed genetic regulations for the behavioral patterns. Vitor, hi. It's never too late. Never too late. How was your day? Any favorite behavioral characteristic you are interested in? This is just the first thing I'm coming through. But then there was a. Oh, actually, maybe this is the. Maybe this is the part I was pondering on. Well, it seems to be the end of the paper. So let's see how they end the paper. Well, because there is not really a conclusions here, so hopefully it's like there will be some illuminating parts here. Anyway, reconsidering the relationship between genes and behavior, Mendel's pioneering studies provided the foundation of the science of genetics. His choice of traits with simple inheritance eventually led others to adopt the genes for shorthand for an expectation of a direct, specific and causal relationship between the gene and trait. Oh, nice. This is actually exactly the thing that I was thinking about how this works. So anyway, so for behavior, it is important to acknowledge that this perspective has been all too easily misused to promote inequities, racism, and genocide. Well, even with the, well, I don't know which is the I don't know if I should click this citation, to be very honest. The studies reviewed here highlight that the relationship between the genes and the behavior is more complicated than suggested by the genes for shorthand words. Good. Okay, so it's as expected, but yeah, so it's nice written, honestly. The things that I read in this paper are really well explained. To be sure, there are indeed excellent examples of individual genes that have DNA variants with a specific and causal relationships to a behavior, some of which affect the expression of many other genes. Okay, Victor, so right now I've been very interested in psychopathology and PGSs. Yeah, it's good to hear. So this topic for me is very new. I was just kind of picking this one because it's just I don't know anything about it. Hi, Isabel. How are you doing? How was your day? Okay, so if anybody has a favorite behavioral characteristic they want to look at, or at least for the possible genomics genomics of that behavioral characteristic, let me know, and we can see if we find papers for that. Anyway, but the studies described in depth earlier have as example as gene for thinking is misguided. It's good to hear that you are awesome, Isabel. Yeah, all good here. There are no genes that specify behavior rather than genetic variation acts to change biochemical and cellular pathways that alter neuronal circuits to result in behavior variation and ultimately behavior evolution. What? Okay, this is the complicated sentence. There are no genes that specify behavior. Well, that's an interesting piece of information here. That is the thing. Because there is a question, so from Isabel, that do these studies take into consideration the indirect genetic effects of these traits? Honestly, I don't know. So this study is the first one. First one is the Mendel's. What is the title? Behavioral Genetics and Genomics: Mendel's Peas, Mice, and Bees. So it seems to be. It's a kind of seems like a summary paper of sorts. It's not really well, to the things that I have seen, it's not really a review, but it's kind of it's very descriptive. That's quite nice. We didn't read the whole thing, or I didn't read the whole thing, but well, just kind of clicked on this one as the first one because one, it was quite high on Google Scholar, and the other thing is that yeah, just interesting title, but it's kind of a nice kind of start to all these things. Although, well, I don't know how much in general I can spend on this, but no, otherwise it's quite fine. Victor, I'm happy to see that you are liking the content. So I think also that is quite interesting, and what I think in general is that it's good to see, good to check out sometimes sometimes the things that we are not really familiar with, and I'm absolutely not familiar with behavioral genetics, and that's why I thought it can I can read something about it. Anyway, so that was the thing here. We were here. So there are no genes that specify behavior. Rather, genetic variation acts to change biochemical and cellular pathways that alter neuronal circuits to result in behavioral variation and ultimately behavioral evolution. So it's also behavioral change. Because most genes are pleiotropic, changes in any one gene often affect many traits. Yeah, so this is what pleiotropy means, that one gene affects more things. Moreover, even the most fundamental elements of a gene regulation are not fixed, and we have as yet little knowledge of the dynamic and the context dependent cause that govern the operation of behaviorally related GRNs. In addition, human genome-wide association studies reveal that associations between specific alleles and behavior variation are heavily dependent on populations that are studied. Okay, so this is also like the effect of the environment then. The genetic background in which alleles are found. Genetic background, I would say more like social background, okay. Providing another example of the context-dependent connections between genes and behavior. Given that behaviors are the product of both nature and nurture, it is clear that both inherited and environmental influences affect the precise and ever-changing relationships between genes and behavior. Figure 4. Undertaking the complex relationship between genes and behavior and how these connections may vary among individuals, populations, and species is a grand challenge for both science and society. Studies such as those reviewed in the pursuing scientists to move past the outdated genes for paradigm, and it's critically important for scientists to work hard to enable the public to recognize and understand the underlying science. Well, it's quite challenging anyway. It is in this way we can honor the legacy of Mendel's pioneering discoveries, highlight the scientific and societal progress we have made since, and identify the important work that still must be done. Okay, so this is in a layer of conclusions, more like a summary, but yeah, pretty nice. Basically putting it that this is basically how the genes might be working in this context. Okay, nice. This is something to maybe check out more. Very nice paper. Next. Actually, this was in animals, but it was more like a general stuff. Okay, so genetics and genomics of animal behavior and welfare, changes and possibilities. Functional genomics of neural and behavioral plasticity. It seems complicated. Behavioral genetics. Organism perspective. Okay, so there is a in chat. Behavioral traits are often measured as ordinal or categorical. Will this be good approach to analyze these traits? You mean by as a categorical trait, behavior, like analyzing the behavioral traits as categorical, so like good and bad, and well, maybe it's not very good but it's basically that putting them into distinct categories. So it's as opposed to like a scale, because I think it's more like I think most of the behavioral characteristics are very much quantitative. So it's like the kind of reactions to something or towards something or against something. It's I think very much a quantitative trait. I think it's something similar, although this one is quite nice, but it's like, I think it's similar. Scholar. Temperament. This is what I mean, right. Human temperament as well. I was more in like I had more in mind the temperament, for example, in dogs or horses, but yeah, why not? Human temperament is also just as good. What I would be interested in if this is or how they, because but if we talk about behavior, I for some reason I always, the temperament comes into my mind. So it's not a behavioral trait per se. I think it's just a kind of a strength of reactions, but it's to me it's always like how strongly the people feel or the animals react or whatever is the actual question. So anyway, so it's like I wanted to get to temperament at some point, so maybe this paper is also as good as any for checking it out, how they maybe also checking on the methods in this case, that what are the things that they are using. Okay, so okay, the title, Uncovering the Complex Genetics of Human Temperament, Sweer et al., with lots of co-authors, which is not very surprising, being in Nature. So I guess it's a lot of people collaborated, and even more people wanted to be on the paper. Anyway, so experimental studies of learning suggest that human temperament might depend on the molecular mechanisms for associative conditioning. Now this is problems. What is associative conditioning? That I don't really know. Okay, but which are highly conserved in animals. Maybe I feel I need to know what is this, because I honestly I don't. Condition meaning. Associative learning is a form of conditioning, a theory that states behavior can be modified or learned based on the stimulus and response. This means that the behavior can be learned or unlearned based on the response it generates. Basically, this associative conditioning is then means that anybody can change the behavior or learn to change their behavior based on their observations, how their environment reacts to it, if I understand it correctly. So, well, okay, so this is basically the associative conditioning, is that basically we can change our behavior if it's necessary or if it's better for us. Anyway, so again, which is highly conserved in animals. Okay. The main genetic pathways for associative conditionings are known in experimental animals, but have not been identified in prior genome-wide association studies of human temperament. Okay, so we use data-driven machine learning. No, I'm just sorry. I'm just laughing a little bit because there is just you know, the keywords and the current or at that time current thing must be everywhere. But yeah, so why not? We use the data-driven machine learning method for GWAS to uncover the complex genotypic, phenotypic networks and environmental interactions related to human temperament. Nice. A discovery of sample of more than 2,000 healthy fins are identified sets of single nucleotide polymorphisms that cluster in within particular individuals. So I mean, just now again, I'm a bit. It's funny for me, somewhat, just because I mean, it's the maybe it's, you know, like it's a bit of a this harmful stereotype, but the Finns are usually depicted as over. Let's put like overly calm people. If I am, well, this is my memory, sort of. And they are used for a temperament study that's just cheeky, a bit funny. But anyway, okay, in the chat, yes, it's basically the classical behavioral models in Pavlov operant condition. Oh, okay, so this is the thing. So it's like the associative conditioning is just yeah, basically what they talking here, the experimental animals, basically the animal studies that they you know, try to do, that they are dealing with the people, or sorry, the animals, and then so that they do certain tasks. Yes. So honestly, the fact that they decided to do it on the Finnish people of all possibilities, I mean, well, of course it's as good as any, but yes, well, why not? Anyway, okay, so they did it with the study. They identified sets of single nucleotide polymers that cluster within particular individual SNP sets, regardless of phenotype. What? I feel I don't understand this sentence too much, but machine learning will uncover the complex genetic environment related to human temperament. In a discovery sample, we identified sets of single nucleotide polymorphisms that cluster within particular. Okay, so there is probably some SNPs that are appearing in certain people, I guess, with certain temperament characteristics. We will see. The second, we identified 3 clusters of people which with distinct temperament profiles measured by the temperament and character inventory regardless of genotype. Well, there is a temperament and character inventory. That is a new information. Third, we found 51 SNP sets that identified 736 gene loci and were significantly associated with temperament. That's quite a lot. The identified genes were enriched in pathways associated by associative conditioning in animals, including these pathways. 74% of the identified genes were unique to a specific temperament profile. That is interesting information. So it seems that it's like, well, there is always a bit of a, I think within these studies, it's always a bit of a danger that somehow the threshold is too low and there is just too many things being quote unquote discovered. Yeah, but it seems, but here if large part of these things was unique for a certain temperament profile, then it might show that these guys are up to something. So, well, but again, just what is from the previous paper, that is like these are not absolute determinants of the behavior. So this is well, the one thing is, well, another thing that is here is the temperament is the kind of the phenotype. So it's by definition, or I think by definition, a quantitative trait. But it seems that it's that these many genes are basically genes of a small effect that influence the quantitative trait of temperament in some small way, if we can. And I think Isabel, also for your remark, I think this kind of proves the the thing that it's I mean, well, maybe proves it's a big word, but kind of this hints into direction that the trait or the behavioral trait should not be classified as categorical, well, to my opinion. Anyway, so and because one other thing is, of course, the environmental factors, which is always we should always consider them. The environmental influence is measured in childhood and adulthood, had small but significant effects. Okay, small. Okay. Okay, so it's like sure, genes must make very small contributions individually. Yes, exactly. This is what I was also thinking, that along the same lines as you, Victor, that yeah, these are the genes that were discovered. That's also one thing. That is, there could be many more that are contributing in a small way. So yeah, it's just interesting in general. Anyway, so but one other thing is that it seems that the childhood and adult, so the kind of this is, I think, about family history, like whatever environment the person was growing up, or well, maybe not family history, but general, just childhood and so on. But what this one says, that it has an effect, but not so big in terms of for the temperament. Okay. We confirmed that the replicability of the 51 finished SNP sets in healthy Korean and German samples, as well as their associations with temperament. Oh, so they cross-checked within Koreans and German people as well, it seems. The identified SNPs explained nearly all the heritability of The identified SNPs explain nearly all the heritability expected in each sample. That is rather surprising. And the one thing is, I don't know, is this is the heritability or in percent, or this is the explained variance type of stuff? Because it's like one thing that if this is if this is the explanation percentage, this is not nearly all for sure, and if this is the heritability, then it's like just far too high, I think. Well, at least according to my expectations, I would say that heritability should be much lower. But again, with a very limited knowledge on the behavioral genetics. Anyway, so explain nearly all heritability despite variable cultures and environments. Okay, that we conclude that the human temperament is strongly influenced by more than 700 genes that modulate associative conditioning by molecular process for synaptic plasticity and long-term memory. Okay, I don't know how the memory comes in the last second, but anyway, so basically they are just reiterating that it's like these genes are important, and there are many. Okay, so let's see what you got. So here also then I'm just interested also in their phenotypes, at what they are dealing with. Okay, I bet is the proportion of the variance explained, but that's for my nearly all. Yes, exactly. This is what I was thinking about as well, because it's well, one thing is it's kind of strange to put heritability in percent in this type of paper, even without the H square or something. And it's like nearly all 35 to, it's like about more like maximum half is not nearly all for sure. So there is an interesting play with words here. So yeah, no, it's, yeah, just a bit confused how these two things come together. But maybe I'm just too stuck on this one issue. But yeah, anyway, but I mean, don't get me wrong. It's like explained variance on the temperament. Like 53% is a lot. That's for sure is a lot, but it's not nearly all. That's what I just wanted to get to. Also, side information, I have learned also some new thing from the last stream. So I have a cup of water with me. So last time I noticed far too late, and then I just didn't want it to go away. But yeah, now it's learning. At some point, maybe I will have a camera as well. Let's see. Okay, so table 1, description of the sets associated with temperament. Okay, that is not so interesting, at least from, well, there is a very large p-values, although percent coding is what? I hope there are some footnotes. SNP sets are named after molecular pathways. Percent coding indicates the percentage of protein coding genes. Strengths of association are compared with the SNP set, the best SNPs and average SNP-based SCAT p-values, the number of subjects and the SNP comprising of each SNP set is specified. The probabilities of the well-being and ill-being, quite nice word, well-being and ill-being, are given for subjects in each SNP set. Character indicates the association of the set with the character phenotype published elsewhere. G indicates the number of genes mapped in the SNP sets, where genes can be mapped by more than one SNP set. Okay, so this is numbers for everything. And this is like oh, and these are the SNP set names. Okay. Sensory sensitivity, conditioned impulse. It's like well, this I think is far too advanced for me to know what is the actual thing, but just generally have an idea. So there is basically the percent coding is for the reason for that, I think, is that they have these regions and there is a bunch of SNPs there, and so it's like this is the number of SNPs I think here. But not all of them might have like an explained function, or there might be some regulatory genes and not coding genes. And this is like just having an idea that how much is the protein coding part, and just kind of scrolling through. Well, again, but that's the next thing is the fact that it's protein coding in that region, it doesn't mean that it has to do anything at all with the temperament or these behavioral characteristics. It's just there. I mean, is it a coincidence? Is there? Maybe yes, maybe no. So that is, if some of these genes, I guess, is they are doing something for the temperament. I definitely think it's so I think that they definitely not all of them. But anyway, just to nice to see that there is these regions seems to be quite well, again, but what is the length of the region? That's also then a question. SNP sets as a temperament. Oh, they are just have it like the, but what is the region? honestly. Because if this is like a half a megabase region, that's one thing, but it's like a 5 megabase region, then it's that's another thing. Maybe. A good thing is to scroll to the methods for this piece of information reference. No methods. Subjects and methods. It's here. Okay. Replicate results. It's very nice that they replicated the results. That's I guess that is also the reason how they get with this high publication here, or high ranking publication in the Nature. It's a nice thing. Also, well, okay. Seems that also there is a gender balance is also quite good. Okay, around 50-50. Well, anyway, so personality assessment. All subjects completed the TSI SS7 heritable dimension of personality. Okay, that is probably some kind of a test. The TCI measures 4 well-validated dimensions of temperament: novelty seeking, harm avoidance, reward dependence, and persistence. Okay. From the chat, with that many SNPs, wouldn't the analysis be underpowered, even if the sample is quite respectable? More than 2,000 people, I don't know. You mean that there are not so many SNPs, or like the because these things are the thing that we have looked before. There are the SNP sets for the so these are the results already. Actually, there is a nice question that what was here genotyped by 670k custom Illumina. It's basically a 600k chip. I think it's like after let's see, after quality control how much remained. Oh, they use Blink. Yes, which is not surprising, but still, if anyone watching this and wants to learn about Blink, for the Genomics Bootcamp channel that you're currently watching is a good place to go, because we have things for basic use and basically the use of Blink for beginners, and also some more advanced topics. Anyway, so after the quality control, but what was the quality control? That was I would be interested in. Well, anyway, maybe it's worth to read this in more. Genotype, the Korean sample, general sample. It's this imputed samples. Okay. After quality control of Blinks, it was used to reduce the large search space by pre-selecting a subset of SNPs using a generously inclusive threshold with Bonferroni correction for possible associations with temperament. Oh, taking gender and ethnicity into account as gender and ethnicity coverage of the individual SNPs as detailed by accompanying paper. We accounted for ethnicity in each sample using the first 3 principal components of ancestral stratification of SNP genes. Nice. Okay, so what happened is they kind of did a pre-selection with a kind of a GWAS type of thing, but they had a very super low threshold. So, but basically they excluded the SNPs. Love Blink made my life less stressful. Yeah, me too, Isabel. Me too. Anyway, so anybody in the chat who was like, if you didn't experience the pleasures of data modification yourself with your custom code, you are missing out a lot. In brackets, you don't missing anything at all, just a lot of pain and sweat. Because the thing is that if you can, this is what I tell everybody whom I meet, especially when I have the opportunity to talk about data. If you can avoid data modification or data kind of yourself, with the custom code and custom script, you should do it. And then this is, I think, the biggest, for me, the biggest asset of Blink. Of course, it does many kind of computation analysis that it's useful, and this is why I use that many times. But one of the big things that it does, it's the just the data modification and reshaping in different formats. By the way, there are also videos on the channel where it's like basically listing all the input and output data formats of Blink. But anyway, so the thing is that it just does it for you in basically one click or one line, and you don't have to spend like days and week on it, and then later on finding out that there is something wrong, that it's kind of comes up with the data. So yeah, this is really good. And the other thing is that because Blink is so widely used, what I love about it is that it's basically an input data format for other software as well, like ped and map files or binary ped files, or there is a Blink 2 coming, and there is also there is a new data formats that coming with it, but also these are could be used for some software as an input, and yeah, just kind of frees up time, so that we don't need to spend things about like we don't have to need to spend time on the data modification, but we can do something else. For example, reading papers on behavioral genetics. Anyway, but the thing is, here I have to be critical, honestly speaking, on this part, because they are well, they are quite short on terms of details, that how many SNPs remain, I mean, I would love to see that. So maybe there is somewhere else, but it's like, well, I feel I don't have to, should not dig for this kind of basic information. But anyway, so the basic question that I had is kind of answered on here, that basically they did this test, and they were the 4 phenotypes. These behavioral things, where is it? Okay, now I lost it again. About this risk avoidance and so on. Maybe I just try to okay. Avoidance. Harm avoidance. Yeah, here, novelty seeking, harm avoidance, reward dependence, and persistence. These are the 4 things that they were looking at. Okay. So, what is the thing? 3D graphs. Not so much fan of these. 2 examples of SNP sets that are represented in a heat map, submatrices, and big clumpers. What? SNP sets were identified in distinct patterns, molecular features. Honestly, just way too complicated, or it's just getting too late. The types of genetic variants mapped by SNP sets associated with temperament, a specific molecular consequences, supplementary, and their subtypes. They have an awful lot of things in the supplementaries. It's a bit, well, it also shows that it's a big study. Multiple SNPs, description of human profiles. Okay, let's see the subtitles. Complex Genotypic-phenotypic relationships in temperament profiles, relationships among SNP sets with one another molecular processes, heterogenic pathways, complex genotypic-phenotypic relationship influence health status, replication of results in 2 independent samples. This is probably the German and the Korean stuff. Estimation of heritability and environmental influences. Well, I hope it's in the let's see. Estimation of heritability and environment. The heritability of temperament controlling for outliers was estimated as what? Is this a heritability? They give it in percent. That's. But then this was indeed the heritability. Let's see the. What is this S21 supplementary table? It's downloading something. Come on. In addition, 87% of the SNP sets were strongly associated with empirical temperament. In other words, the SNPs that comprise the different SNP sets strongly distinguish the temperament features of the subject in each set, indicating that each individual SNP set contributed significantly to explain the total distributed heritability. Consequently, when then the genotypic sets were used to classify the well and ill-being of the subjects using the PGMRA classifier, predicted values were highly accurate, average, etc. Yeah, so this I'm also indeed surprised about the if honestly temperament heritability like 0.5. I mean, it's like, honestly speaking, it's not so much of a difference between 48 and 53%. But I would say like around 40 is also very, very high. I would be expecting, yeah, I don't know, below or around or below 0.1, honestly. Discussion. It's just a far too long paper with a lot of stuff. Strengths and limitations. Let's see what it is. Molecular pathways. That's I'm not super interested in. Strengths and limitation. Major strength of his finding is the strong replicability of the findings in 3 independent samples from different cultures and independent studies of gene expression during behavioral conditioning for experimental animals. I had a. It's not people. Oh, the animals come in with the independent studies of gene expression. Okay, so that the gene expression was found also in experimental animals, and they were confirming it with 3 independent samples. There are 2 different things in this sentence, and this is also one of the earlier criticisms that these authors should write more sentences. Because it's like this way it's confusing, honestly. It's like this is one side of information and this is the other side of information. I would like to really much to have it separate, so it's not confusing. Anyway, but it's very, very nice. The one thing is yeah, indeed, Victor, it's not the variance explained, which is honestly a surprise, just a general surprise. But anyway, so yeah, so the replicability of the findings, it is especially in GWAS, is one of the big issues. And the fact that they did not just one or but two independent sets of nationalities, then yeah, then it's that's a good thing. Yeah, so there's actually, yeah, Isabel, valid question of what are the standard errors around those values. Let's see. I try to look them up in the results again, where they are kind of showing them. While the true cluster analysis is hypothesis generating method, which there are no unique solution to the number of clusters with features are relevant for the cluster degree of homogeneity demand for each cluster, included practical and robust solution for each of these problems. Honestly, it's oh, this is already the end. Conclusions and recommendations. We were able to describe and replicate the complex genotypic-phenotypic risk architecture of temperament in 3 independent samples of people. Our unbiased data-driven findings confirm the hypothesis that the temperament is based on associative conditioning. Okay, that's what it is. Basically, it can change. And related processes, particularly stress reactivity in response to extracellular stimuli. We have found that different molecular and cognitive processes are associated with character, but health status depends on the genotypic-phenotypic relations and that influence both temperament and character. Therefore, we recommend further work to examine the overlap in interactions between the temperament and character, which is all nice. Why are you writing this sentence on the end of a Nature paper? Of course, it's not the end of the research, but why should you write that more research is required? Come on. Yeah, don't write this. Like the more, of course, it's more research. Always more research is required, but I mean, but it's sort of refreshing to see that it's regardless of the publication level, the people are thinking similarly and doing similar things. Yeah, but if I see this in a paper of my, well, for example, for my student or whomever, like in my collaborators, I'm always pushing for removing such statements, general statements. Indeed, Victor. Now it's like, yes. It's kind of, honestly speaking, it's a little bit like degrading the results. Is not a good word, but it's kind of make them less than they are. Because I mean, you basically you do the research and present the results, and hopefully these results are kind of solid, and you can show something. And then at the last sentence is like, oh, but we don't know though. So it's like, there is lots of things that we don't know and more research is required. Yes, but it's kind of a almost an apologetic kind of sentence or phrase at the end that is like, why would you do that? Maybe I'm just too I'm just talking way too long about this sentence, but it's just generally I'm against it, as you might have seen. Anyway, standard errors. It would be good to see. That's indeed. I have a feeling that it will be buried somewhere in one of the many of the supplements, honestly speaking. One thing that it's because of the time pressure, standard. No, I thought it's like they are, but the one thing that we can be sure about is that they do not talk about the standard errors of their estimates in the main text, which is a bit well, what is the best word, because I'm on the record here. They could have thought about presenting the standard errors as well. Um, uh, how about simulation papers? What do you mean, Isabel? For the more research is required type of statement, or to me it's that statement, it doesn't belong anywhere. Like, no, it's just no. I mean, it's my personal feeling, or you know, like stance on it, but no, it's just no. Why would you? The same thing, which is some people do, is like that in the title they put preliminary study of this and that. I mean, this is also kind of along from the same basket. Why would you write that it's a preliminary study? It's like that is then immediately kind of also downgrades the study, in a sense that if I would see preliminary study somewhere, I would say, oh, this is not the real study. Why should I read it? And of course you would write a paper so that the people read it. So just don't write that it's a preliminary study. It's, well, of course, again, it's like not the end of the line for sure, and maybe that is the beginning of something, but still do not downgrade your own work by saying that, oh, this is thing that we put a lot of effort in, but it's, you know, not very important, or we feel this is the beginning of the road, so that is like, you know, one small piece in the big ocean. Of course it is, but it's, do not write that this is a preliminary study of some small importance, and you know, just don't. So that is the preliminary study, that's in the titles, and the more research required, absolutely no go. And I stop with this topic here, because it's just then it's also tells about something about my behavioral patterns. Anyway, time is running. It is midnight where I am at in Vienna, Austria. So I think I will stop this stream here. So although coincidentally it's like the one of the most amount of people in the chat at the moment. So folks, I'm willing to extend, but from the chat, from people in the audience, put something in the chat that what should be the last paper of the day to look at. What are your suggestions? We could check out one more, but yeah, something for something, you know. Sunil, hi, just joined the stream. Oh yes, so the stream is going on for some time. Continue. Oh, why not? What is paleogenomics? Friend, that is for some next time. So right now the topic is on the behavioral genomics, so around some traits or behavioral characteristics. Coincidentally, paleogenomics was also on. So there, well, I made a list that what are the kind of these overall topics that could be looked at. And the paleogenomics is one of them. So last time, and you might see also like on the Genomics Bootcamp channel along the live streams. So we did something on archaeogenomics. So it's that is the, well, I know it's not paleogenomics, but it's something in this direction, and paleogenomics might come as well. Cattle aggression and genes. Hello. Sehira? I hope I get the name right. Sehira Farm. Yes, cattle aggression and genes. That is a really nice topic. We might look at it. Yes, so it's kind of along the lines of, actually, it fits quite well because of the, well, it was in the temperament was discussed. I don't know if you were here at that time. So this I think we spent a bit more time on this paper as for others, but it's like the uncovering of complex genetics of human temperament. But yeah, the cattle temperament could be also interesting. Okay, so let's try that. Let's try that. Where are we are at again? Okay, so, and what I will try to do is, when in doubt, and then do the kind of exact thing. So I will just type out whatever you wrote. Cattle aggression and genes. And the genes might not be agree. Is it with double G, huh? Thank you very much. It was very nice. Can we do it more often? Yes, in fact. So what I'm doing, I thought I will be, well, one thing I know that, and actually the videos, there are some that are planned, some that are planned for a long time, but somehow they are happening more slowly than anticipated. So thank you for your patience, definitely on those things. But still things are in plan. But so one of the more convenient types or things on this live stream that basically, well, it's just happening. So the one thing is the editing is well, there's still preparation and stuff, but it's, at least I don't need to edit. Although what I was actually thinking that I might do then a cut, kind of a highlights for this, for also from the live streams at some point. But anyway, so yes, we'll do it more often. I don't think it's this week because it's like then it doesn't fit very much the schedule. But yeah, from time to time, definitely. And I will try to. It seems that there is a bit of an audience of the, and yeah, thank you, Victor, for the. So yes, the format is quite interesting, and. Yeah, let's try, and I will try to put up a schedule, because I mean the last one was absolutely unannounced, and this one was announced on Twitter and LinkedIn like in a very last minute. So just basically just before the stream started, like that's an hour ago. So maybe I will try with a bit more kind of fixed schedule, and then also you will know that what is the thing. And meta founders and unknown parent groups, that's rather specific, but like I make a note on the meta founders and parent groups, and we'll see what happens anyway. So but coming back to Zahira. Yes, so that we are here. Gene expression profiles underlying aggregate prefrontal cortex of cattle. Aggression behavior cattle associated with polymorphism, gene promoter. Genetics of behavior in cattle. From Slovakia. Genetic selection signatures, agnostic breed, genetic control, dairy cattle behavior. Anyway, so let's see. Let's limit our things here a bit, and let's see our options. So gene expression underlying aggressive behavior. So this is one thing, and this other one is because you told genes, and then, well, there is this one gene here for sure. RNA-seq, I would age effect. Oh no, this is something gene expression pro and underlying prefrontal behavioral traits. Oh, this seems to be nice, at least from the title. Anyway, so there are 3 papers. So let's see them. Hopefully they are not for to be okay. So it's open access. Anyway, so the first paper, and what we'll try is really nice to kind of go through in a more express way in terms of abstract, subtitles, pictures, conclusions, so that we also get through things more quickly, because yeah, I mean, it was an interesting discussion, but just 2 papers took an hour. So so anyway, so gene expression profiles underlying aggressive behavior in the prefrontal cortex of cattle. Authors Eusebi et al. From, I don't know from where, but for sure it's anyway. Background. Aggressive behavior is an ancient and conserved trait habitual for most animals in order to eat, protect themselves, compete for mating, and defend their territories. Genetic factors have been shown to play an important role in the development of aggression both in animals and humans, displaying moderate to high heritability estimates. Interesting. This is something that is like comes back again, and just surprising for me, because I thought it's like the heritability must be low, but it's like again the next paper that is saying that moderate to high heritability, which is, and that the previously it was also around 0.5, 0.4, 0.5, which is a lot. Although such types of behaviors have been studied in different animal models, the molecular architecture of aggressiveness remains poorly understood. This study compared gene expressions profiles of 16 prefrontal cortex (PFC) samples from aggressive and non-aggressive cattle breeds, Lydia selected for agnostic responses and Wagyu selected for tameness. Oh, this is the probably the this fighting cows or whatever. Thanks for the stream. You're welcome. Pantuflor. Yes, I think this Lydia, just to see the like hopefully we'll not something cows. Yeah, these are, I think, this one of the fighter clans. And Wagyu is the Japanese thing known for their meat. Anyway, so temperament aggressiveness, just to do not get off track here, because that's what I can do very easily to get off track. Okay, so total of 918 upregulated and 217 downregulated differentially expressed genes were identified. Well, again, the same thing, that's a lot of genes. Also in the previous study there was, I think 700 something genes. This is also a lot. It's kind of around the similar magnitude, at least. It's in representing above chance overlap with genes previously identified in studies of aggression across species, as well as those implicated in recent human evolution. The functional interpretation of the upregulated genes in aggressive cohort revealed enrichment of pathways such as Alzheimer's disease, presenilin, well, I don't know what it is, integrins, and the whatever signaling cascade, all implicated the development of abnormal aggressive behaviors of neurophysiological disorders. What? Okay. Moreover, gonadotropins upregulated natural mechanism enhancing aggression. Oh, so basically they compared the hyper aggressive ones and the very tame ones, and they got the genes, and they basically what they found is indeed things that were connected to aggression, which is nice and logical, but it's nice to know it worked, because it's not necessarily coming out all the time, but it seems that this time it did. So concomitant lean have just come way too complicated. Sorry for my English, but it's like the words are way too complicated in this paper. heterotrimetic G protein pathways associated with low reactivity mental states, and GAD2 gene, the repressor of agnostic reactions associated with the prefrontal cortex activity, are downregulated. Also, the repressor of agnostic reactions is downregulated in the aggressive ones, which is again logical, but nice to see that something like this kind of came up. Promoting the development of the aggressive responses of selected in the DDX, promoting the development of aggressive responses of. Okay, so the one thing is the mechanisms enhancing aggression, and the other thing is the genes, the repression of the, or the kind of the downregulation of the genes that repress aggression. So that is, so it's both ways. Both ways are kind of, so the positive genes are, in terms of aggression, are upregulated. The negative ones that would lower it are downregulated, so they are less. Promoting the development of aggressive responses selected in Lydia cattle. We also identified 6 upstream regulators for functional activity fits in the etiology of abnormal behavioral responses associated with aggression. Okay, so the genes is the GAD2 for the repressor that should go down, and there was one more, or some others that are conclusions. These transcriptional correlates of aggression resulting at least in part from controlled artificial selection can provide valuable insights into complex architecture that underlies naturally developed agnostic behaviors. This analysis constitutes its first important step towards the identification of genes and metabolic pathway, promote aggression in cattle, and providing novel model species to disentangle the mechanisms underlying variability in aggressive behavior. Okay, good. So it's they have genes on that methods. Let's see, cross species. Do they have pictures? That's the question. Okay, log fold, like downregulated and upregulated. Showing this MA plot, this would be this, showing the distribution of differential expression genes. The y-axis shows a log 2-fold change of expression between aggressive and non-aggressive groups, and the x-axis corresponds to log 2 transfer average expression level for each gene across the samples. Hi, bye Isabel. Have a nice evening or day. But we have been again. So they in the chat. But we have been breeding domestic cattle for docile since 100 years ago. So it should be less aggressive than a wild cow, isn't it? Yes, that's definitely so. So in general, the temperament in cattle is crucial because, well, nobody wants to be crushed by their cows. But there's still a variability, honestly speaking. So in any population, you actually can measure temperament and find different kind of more and less aggressive individuals. But this is a very specific thing. So this is a GWAS study on aggression, basically, or in cattle. And what they did is, in this case, it's always good to have like a case and control. So basically, extreme parts of the. So basically, the two sides of the coin. And in this case, what they did is they went with a cattle breed. That this one is, I think it's. Well, I'm not very familiar with it, but it's basically about this fighting. Behavior is encouraged also within the selection itself. So this these are bred for agnostic. So, as it's like written here, selected for agnostic responses. Basically, it's that they bred in a way that they should be very temperamental and aggressive and pushy and so on. And the Wagyu is another side which is like kind of known very much for its meat quality and so on, but also it's like a very docile nature. So this is what they did, is that basically they compared the two extremes, and based on this comparison the things are coming up here. So, and the reason for this is that so that they can find actually the genes that they have to do something with the aggression in cattle. That's how the thing is. So in general, you're right that it's like the cattle is more tame than it was at the time of its domestication, but still this particular study is about finding the genes that actually do something in the aggressive behavior. Partner and which pathways? Honestly speaking, I'm not so enthusiastic about these pathways. For sure, they are nice, but yeah, exactly. Like because of this, I guess this is something like a regulatory network. Well, honestly speaking, if you look at this network, like look at this, what you learn about this one, what you learn about the status or the of the nature or how nature works based on this. I think it's far too much information, and yeah, again, maybe most likely I'm too critical, and I think well, I don't know where it is, but there is also I've seen a picture that was a biochemical processes in the brain or something like that. It's like one of the most, or at least I think it was part of the biochemical processes in the brain. It was a large, super complicated picture. So it's that kind of just to illustrate that the nature is super complicated. But honestly speaking, if I read the paper, I don't know if I have the capacity to understand too many things at once. And if you present me with this, which is then everything is connected to everything, I am a bit, how to say, confused. So I like simple statements. Anyway, so as I said, and this yes, I get it that these are these pathways and so on, but again, just I don't know what to learn. Oh, now this is a bit nicer picture here, less things on it. that's what I like. Okay, so this would be probably the gene names or actually regulators. Just try to fit it somehow into one picture. Okay, so major upstream regulators of the network of differentially expressed genes in the aggressive Lydia group. There are 5 upstream regulators predicted to be activated, underlined with black color. What are then the black colors? Honestly, none of these are underlined. We activate the 5 group activated, underlined in black color. Anyway, so maybe these ones. 1, 2, 3, 4, 5. Maybe these 5. Okay. In red, upregulated and green, downregulated, we can see the genes with expression changes in response to the activation of upstream regulators. The shapes of the nodes represent the functional class of each gene or gene product as defined in the legend. The straight and dashed line represent the direct and indirect interactions. Yes, so this is what I was. So for example, if you present me a picture like this, it's still quite complicated, but at least there is some so I can make sense of it. So, but so there is the genes that they are, and there is some kind of a pattern that they are connected to each other, which is still very complicated, to be honest. But and these are the various functions and or what is it, functional classes. So they also, they are genes, but they are doing different things, which is also shows, but partly also this is the reason why I'm a bit surprised that the heritability would be even moderate, honestly, and they say moderate to high, because if something is very apparently regulated by many genes of, I assume many genes of small effect, that how come that the heritability is high? By saying this, I just realized that, for example, in cattle, some other traits, for example, everybody's favorite milk production is also 0.3 heritability, and it's also a quantitative trait. From the chat, hi Sunil. So, does genes regulated anger or calmness returned on and off all the time? That's an interesting question. Honestly, I don't know. Seems, based on my limited information, that it's that would be a question for epigenetics. Is it like the kind of changing of the stances, turning on and off? That's I think the answer to this question, if the genes regulating the temperament, basically, if they are turned off, or you're right, if they would be stimuli dependent. Well, in this case, no. In this case, I would be fairly sure that no, at least not on the short term, because if that would be stimuli dependent, for example, there would be, I don't know, like a thunderstorm, for example, that would kind of provide a stimuli for fear reactions. I don't think this would be a kind of a thing that it's that would just, it's even possible that that anything is changes on the genomic level or genetic level. Well, in this case, genomic level for that. So in this case, definitely not. The question, for example, if you would put, for example, an animal into an environment that would be very much extreme in one or other way, extreme calm or extreme stressful, by the end of its life, or kind of that, so if there would be some epigenetic changes or turning on and off genes, that's hard to say. Honestly, I don't know. My feeling is that maybe. The answer is a strong maybe. But I could imagine. Honestly, I could imagine that that such a thing would happen. But I have absolutely no proof whatsoever. So that would be my take on this issue. But honestly, for the short term, no. But for the long term changes, it's a very interesting question, and I don't know the answer. Okay. Okay. So this is not. Let's see. This is a long discussion, and they're fairly long conclusions. But let's see. This is the first time that a comparison of the differences in genomic expression between aggressive and non-aggressive selected cattle breeds have been performed, identifying a lot of genes, up and down regulated. We have also undertaken a cross-species comparison of analysis to identify genes in common implicated in aggressiveness and to investigate their regulatory networks. This is what we skipped in the paper. Our results indicate upregulation of the aggressive cohort of pathways such as the Alzheimer's disease peresinilin. This is this complicated words, integrins and park signaling cascade. All routes implicated in the development of abnormal aggressive behaviors and physiological disorders, even disorder. Also identified normal mechanisms enhancing aggression across species, such as the upregulation of gonadotropins and hence testosterone, whose levels have been widely linked with the agnostic reactions. More testosterone, more aggression. Well, makes sense biologically, honestly. In the contrast, heterotrimetric G protein pathways previously associated with low reactivity mental states like those involved in a major depression, or the GAD2 gene, which plays a pivotal role in the control systems deployed in the by the this I always forget what is this to repress agnostic reactions, are both downregulated, guaranteeing the development of the adequate combative responsive needing during the corrida event. Oh, this is the corrida thing, is the when the I think the cows are fighting in there. So it's like the top cow, most aggressive cow. I think that's the thing. This is, I think, the corrida. Nonetheless, despite the PFC being a key region for the modulation of aggressive behavior, it may not be representative of other brain regions also reported to play important roles in aggression, such as the amygdala, hippocampus, or hypothalamus. Furthermore, although our findings are consistent with the gene expression studies for aggressive behaviors in other species, one limitation of the current study is that we still need to consider some inherent biasing factors, such as those associated to breed differences and external factors such as conditions and of the animal's slaughter. Okay. Nevertheless, this study constitutes an important first step towards the identification of genes that promote aggression in cattle, provides novel species as model organism for disentangling variable mechanisms underlying aggressive behaviors in our own species. Also, a neat tie back to the humans make the article that more relevant. Nice. Honestly speaking, nice thing. So there are some genes mentioned as well, and in particular so it's like focusing on this prefrontal cortex. Not bad at all. Okay, so just look briefly on the other two. It's the same author. Eusebi, Paulina. Eusebi, same authors by coincidence. Not all of them, but aggressive behavior in cattle associated with the polymorphism is the MAOA gene promoter. Okay, in this case we just take the abstract, to be honest, because it's like okay, so molecular mechanisms underlying okay, so the thing is the aggressive behavior in cattle. Molecular mechanisms underlying aggressive behavior are primitive and similar among the Cyphum vertebrata. Nice sentence. In humans, a primary goal of the study of aggression is to determine the neurobehavioral molecular factors in triggering violence. Although several species have been used to study agnostic responses, researchers are limited by the difficulty of artificially inducing aggression in animals not selected for it. Conversely, the Lydia cattle breed has been selected since the 18th century to display agonistic. I read it wrong all the time. Agonistic responses based on the traits such as aggressiveness, ferocity, and mobility, all of them showing significant heritability values. This intensive selection may have driven shifts in specific allele frequencies. In previous analyses across the autosomes, we revealed the long-term selection regions, including genes involved in behavioral development. In the present study, we focus on mapping recent signatures of selection associated with aggressiveness at chromosome X by comparing Lydia cattle samples with two non-specialized Spanish breeds showing tamed behavior. It seems that this Lydia cattle is their favorite, well, for good reason, honestly. The most significant markers peaked around the monoamine oxidase A, MAOA gene, and thus associations of three functionally important regions located near the promoter of this gene were further investigated. The polymorphism consisting of variable number of tandem repeats of the nucleotide C with this BTX, so it's X chromosome on a X chromosome is so big I didn't realize. So this is 105 megabases, 105.5 megabases on the X, and displaying lower number of repetitions in the Lydia breed when compared with the tamed breeds. So it was detected, the lower number of repetitions were detected in silico analysis. In silico meaning that in a computer with the genotypes predicted that the G, well, 105 million, etc. C variant may code for the SP1 binding motif, one of the major transcription factors controlling the core promoter and expression of the MAOA gene in humans. Good. Nice. Again, honestly speaking, this is also their thing, is like they're doing something on aggression on the Lydia cattle, last sentence tie back the things to the human research to make it more relevant. Nice strategy, seems to be working. And also I think well, I don't know how frequented is this field. Well, the paper is not the newest, but still nice. And third one, I hope it's not for the same authors. No. Yeah, so this is from behavioral traits in Bostaurus cattle, their heritability, potential genetic markers and associations. Okay, so let's go for the abstract. Okay. Now one more, just because I started just one more thing in here. So I like that how in the half of the summary until here, until this is basically a big introduction of the things that what is in just the general stuff, and here they start in the present study. So it's like the half of the summary is not about the present study, which is no. I was reading, I was kind of thinking that when they want to get to their stuff, but it's like probably it's a thing that they wanted to communicate is this is MAOA gene. So, and that's what they did. So, but yeah, so that was quite funny for me that they spent a lot of time in the summary, or a lot of space by just setting the scene, so to say. Okay, and the last one, just clicked by, so Francis Margaret Titterington et al. From UK, it seems, Hillsborough. Okay, let's see what you have to say about the paper, behavioral traits in Bostaurus cattle, the heritability, potential genetic markers, and okay. So let's see that if they say something about the heritability. It was teased many times, but I haven't seen it, at least in the previous papers. Okay, people who work with cattle are at a severe risk of serious injury due to size and strength of cattle. Yes, the risk can be minimized by breeding less dangerous cattle, which have more favorable reactions to humans. Also, yes. This study provides a systematic, this is a review of literature pertaining the cattle genetics relating to behavior. Review protocol was developed using the preferred reporting items of systematic reviews and meta-analysis. This is a PRISMA framework with population, exposure, and outcome components identified as bovine genetics and behavior respectively. Well, I am missing a lot of information here what this means, but I take the word of the authors of it that it makes sense. 49 studies were identified in the shifting and assignment non-exclusively to groups of heritability, 22, genomic associations, 13, and production traits related to behavior. Behavioral traits were clustered into following groups: temperament, disposition, and or docility. Another one is aggression, shoot score, I don't know what it is, flight speed, milking temperament, non-restrained methods, and restrained methods. 14 papers reported high accuracy, standard error less than 0.05, estimates of heritability, and the majority of these studies measured over 1,000 animals. This is nice, honestly speaking. That is like high accuracy, high number of animals in the study. This is in 12 studies. That is really, yeah, this seems to be a quite nice review. And if you are interested in this topic, I think this is maybe one of the places to start. Well, just the reviews are nice to read in any case. Like, yeah, so it seems to be quite a collection here. The heritability estimates were found to vary between studies. Well, I'm just wondering how much. So many teasers and nobody tells what it is. Gene associations with behavioral traits were found on all chromosomes except chromosome 13. Strange. In associated SNPs reported on all chromosomes, all chromosomes except. Okay. Generally, it was found that the correlations between behavior and production traits were low or negligible. Okay, so correlations between behavior and production traits are low or negligible. So basically, how the animal behaves doesn't tell anything about the growth rate or milking potential or milk breeding values. These studies suggest that adaptive improvement of behavioral traits in cattle is possible and would not negatively impact. Oh, this, yes, actually, that's a nice. I was not thinking about it this way, but it also seems that it's the one thing. Come on. So one thing is that it doesn't. The two things are not connected too much. It seems. So that it means that you are quite free to select animals that are more docile or more non-problematic without impacting the production, which is great. So basically you can have better behaving cattle and still the good production. However, the variation between studies demonstrate that genetic relationships are population specific. Thus, the assets of heritability, genetic associations with production, and genomic areas of interest for behavioral traits, a large-scale study of population of interest would be required. Again, this more research required. Ending. Yeah, come on. Anyway, but it seems. Why? No, I'm not getting into this again. But why? Why would you say you did a quite nice collection of papers? It seems that you say, oh, but bigger thing would be required. Yes. Why then it's yeah, just doesn't matter. Okay, so let's see. Population what I want to find, hopefully, and if there is things on the heritability, behavioral traits. What is this? Study selection, temperament and docility, aggression, cute score, milking. So these are the things that they heritability. Okay, and this is the thing that I'm interested in. I'm just let's see what it is here. So behavioral traits grouped by trait category in italics, considered reported high accuracy heritabilities. So temperament, disposition, and docility. Temperament and standard error. This is a breed, aging test and reference. So there's quite nice. Anyway, so temperament, low heritability. Farmer assess temperament also. Why they say then is that moderate to high heritability if it's like not so not so well, I would not say so score. Choose score, yeah, Vitor, I also am wondering, but I haven't. If you look it up and if it's something illuminating, then if you could post explanation on the chat on that, would be great. Okay, so temperament also lows. Why they say that? Okay, so there are some higher values here, but in general, temperament seems to be a lowly heritable trait, so it's not that surprising. Well, there is some difference between the. This is some. But it's anyways like around 0.1. This is more the values that I think I would be expecting kind of. But it is again in heifers 0.26. This is docility. Now there's a question: what is a difference between the docility and the temperament? To me, it's quite the same. But there is quite a variation in the heritabilities, it seems. 46.6, that's a lot in Limousin cattle. Actually in the same Angus. What is this? Welcome. Come on. From 2016 from Australia. So it's a lot. Okay, so in the choose score we have a input. It is so the choose score is it's in a 1 to 6 scale. It is the rating of the cow's aggressiveness when it's being held in a chute. 1 docile, 3 restless, 6 aggressive. How high value of the trait should be to consider it heritable? No, it's Sunil. Good question. The thing is there is no threshold. So basically, there is basically any phenotype that you can measure or see, and if it is basically the heritability is nothing else than the proportion of the genetic variance from the overall phenotype. So if it has like a genetic background and you know, like part of the genome that influences it, and basically if the parent and an offspring are kind of more closer to each other in terms of the expression of that trait, then it's generally it is heritable. So there is not a question if there should be a certain kind of threshold in any trait or measurement or something so that it should be heritable. The heritability coefficient is basically then the proportion of this genetic variance. So I mean it's ranging from 0 to 1, and 0 would be non-heritable, and the 1 would be, or 100% would be that is absolutely heritable, and there is no environmental influence on that trait whatsoever. Most of the traits are none of these two extremes, but it's somewhere in between. And the thing is that if it's like 0.1 or around 10%, so then it's well, I mean, technically it's not the proportion of explained, but it's the variance of that trait is influenced in 10% by genetics and 90% by environment. So it's not that. So there are some particularities here with the interpretation, but the kind of long story short is that the 0.1 is a rather low value in terms of that it's heritable, but it's not influenced by the genetic background so much. For example, other traits, for example, milk production is around 0.3. That would be, like a slightly higher heritability. And for example, in cattle, let's say the how to say, shape traits or the this muscle not muscle mass, but there's like this slaughter traits. Some of the slaughter traits have rather high heritability of 0.7 or something. So that is kind of saying that is like influenced by genetics a lot, and environment a bit less. And the kind of the thing here, what I'm still continue to be surprised about, is that the heritability of the temperament or behavioral traits in general seems to be higher than I expected. Because honestly, I was expecting around this, or maybe even less, 0.1, but it seems that for some, depending on the interpretation, it can be much higher. That is like 0.67 or 0.4, or it was also even 0.5 something in the previous study. So that was on temperament also, I think. So this is the shoot score. That seems to be also quite. So this reaction of the cattle seems to be also quite heritable, or quite a lot influenced by the genetic background or genes. And some other ones, aggression, it seems to be on the lower end again. So it's like at the end it's not as drastic than I thought. So it's like kind of there is a variation in the heritability depending on the trait and depending on study and depending on the population, it seems. Which is doesn't add too much clarity into the thing. So it's like, but it's still kind of telling the overall kind of message that it's quite highly heritable, honestly. And the other thing which is in this paper, what they told, that the production levels are not so much linked to it, so we can do selection on the temperament. Heritability of aggression, heritability of milking temperament, which is, I don't know what it is, it's milking speed. Genomics background. Yeah, so there is it's a lot of descriptions. Well, it's a review, so it's a long thing. Anyway, conclusions. Search protocol to return studies which describe the heritability potential associations with production or the genomic areas of interest of behavioral traits in Bostaros cattle was developed and returned 2,238 papers. Relevant papers were assigned non-exclusively to groups of heritability, genomic associations, and production traits related to behavior. The heritability of behavioral traits means that the additive improvement of the temperament is possible and beneficial to animal producers. Yes, this is exactly what it means. Correlations between temperament and production traits suggested that the selection against animals that are highly reactive to improve. It's just a complicated sentence. Correlations between temperament and production traits suggested that selection against animals that are highly reactive to improve welfare and the ease of handling would not have detrimental impacts on productivity and reproductive output. Yes, I know what they want to say, but I feel this is just a way too complicated. I mean, just it could have been written in a simpler way. Anyway, so basically it's telling that we can do selection for temperament, not impacting productivity. And was the reproduction also mentioned before? I don't remember. But okay, so from the chat, and they also examine different ages. Some are mature, some are weaning, I wish they reported the actual average age for that. Yes, yes. So I saw that it's like in addition to many other categories, the age was also different. I think it has to do something with the fact that they this is a review, so they collected the studies, but yeah. So I don't know. I don't know if it's possible to report on that, or maybe there was not so many studies that for each age group, or everything was a bit different, so that they could have been categorized together. So again, a question from Sunil: did trait interaction? So if the trait interaction was measured, I believe traits could complement and non-complement each other. Yes. So there is the. So basically, we are talking about. I think it was well, not very much in this context. It's in a similar context, but not exactly. So they were about when the genes were discussed on the temperament. So there was some pleiotropic effects mentioned. So the thing is that it seems that the genes that are influencing all this, they have this pleiotropic effect, meaning that if they change, then they are influencing more than one trait, and then could be that this more than one behavioral trait. So this is what I got, at least from that particular paper. So in a way, then yes, you are right. So it's like there is a interaction, somehow a connection between the traits. If it is directly an interaction between behavioral traits, I'm not sure, because this is a review. So they basically just checked what the other people were telling and summarized it in a nice way. So again, it's a very good question, but I cannot answer it right now. So it might be that there are some papers that are, or may also actually the previous one was mentioning all these, or even the one with the humans, with the samples on the Finns, Germans, and the Koreans. That is like that they were these trait groups, or not trait groups, but what was it? These reactions, or no. So basically these behavioral patterns, and maybe that was also there was some kind of a connections there. So maybe well, if you haven't been here, so maybe then once this stream is over, if you can search back to the first paper, and there were these 4 groups. Maybe check out that paper, and maybe there is something on the trait interactions on that as well. But here I haven't seen, or in general, I haven't seen on that. Okay, where were we? Also, there were genes, and SNP reported the breed and animal type differences between studies meant that the meta-analysis was not possible. Okay, the findings of this paper can inform breeding decisions. Again, it's strange phrasing, although they should be from the UK. And if employed with the suitable management, lead to the improvement of animal behavior and in turn farm safety. What? Lead to the improvement of animal behavior and in turn animal safety. Furthermore, the variation in heritability estimates correlations with production and genomic associations of behavioral traits reported in the studies reviewed highlights that these relationships are population specific and age specific and everything else specific, as we have seen from that paper table. What they in this paper, it's a rather nice summary. There was also found to be variation in management systems and methods of behavior assessment and scoring. Thus, to accurately assess the heritability genetic associations with production and genomic areas of interest for behavioral traits, a large-scale study of the population of interest using uniform comparable methods would need to be required. Okay, so honestly, chat, is my English broken? Is it too late, or is this thing strangely written? Just if you can give your opinion on this. So it's like, technically speaking, so the authors should be from the UK, so I guess they speak English every day. Or maybe it's me who is just too it's too late here, but a large-scale study of the population of interest using uniform comparable methods would need to be required. Okay, maybe I'm just hooking up on the unimportant things, but I have a feeling if well, some kind of or it's probably me. It's probably me that I'm a bit tired now. Anyway, so chat, people in the chat, and also others. I'm happy that you are here. You were here for this event, or well, maybe I don't know if we can call it event, live stream. We did a double time, double time, as it was. Honestly speaking, I was like, oh well, I do it until midnight. But then there was a encouragement from the chat that we should go on, so we went on and we did. Actually, I'm quite happy that we did, because these last 3 papers on the cattle aggression or aggression traits in cattle were interesting. So this was behavioral traits in Bos taurus, the aggressive behavior with this MAOA gene, and this was quite nice, this prefrontal cortex in cattle type of stuff. So that was it was a time well spent, I believe. Anyway, so I just want to thank you for being here. Thank you for your continued support of the channel, also implicitly by being here. If somebody is not subscribed, then maybe now it's a good time to do so. You can press that subscribe button, and maybe yeah. So also it helps to notify if I am online again for the live stream session, and also I will definitely will return also for producing and uploading also the fixed content videos on various how to say that? No, so they're basically tutorials and others. So there are things that they are planned, but you will see once they come up. But anyway, so I quite enjoy these evenings, or like checking out things with you together. So probably we will do something similar for some other topic next time. So if you are interested also on the contents of this live stream, if you by chance haven't seen things, then this is online as of now, so you will find it on the channel. And also the last one, I think it was from Monday, was on archaeogenomics. So that was also a few nice things also there. So thank you very much again, and so we will end it here, but definitely we'll return with a similar format. The soonest the most earliest next week, to be honest. So, and then yes, I will post then. Also, well, you find my Twitter, so that also I will post things there, so that so the announcements of the streams and so on. So thank you very much again. As of always, I definitely thank you for your time you spent here, and have a very nice evening, have a very nice day, whatever your time zone is. And yeah, so we see each other next time. So thank you very much, and have a good night.